So on plate 1, kind of take-home messages is review the organelles. We're going to spend time talking about the mitochondria and its role in energy production, oxygen consumption when we talk about aerobic and anaerobic muscle. Lysozymes, so the lysozyme. Vacuoles containing lytic enzymes, enzymes that lyse or disrupt. They play a very important role in defense against bacteria and viruses by melding with the invading proteins and lysing and destroying them. They are also fundamental in the meat science process, as in muscle cell death, the lysosome membranes break down. They release these enzymes that start an auto-digestion of the cell. So under a normal functioning cell, this membrane is intact. These enzymes are restricted to the organelle, but in death, they get released and they produce a digestion or lysis of the muscle tissue itself. That produces a tendering of the muscle. So if you would go through and review the cell structure on plate 1. Plate 2 talks about epithelial cells. Most of your focus on epithelial cells will be in the laboratory, so I'm not going to take information from this plate at all. Plate 3. DNA replication and cell division. Basic biology. Again, this should be reviewed. Interphase, and then the processes of mitosis and meiosis. We'll come back and talk about those processes in a little more detail when we talk reproduction physiology. But again, I want you to review now the phases of mitosis, the prophase, metaphase, anaphase, and telophase. Plate 4. DNA expression and protein synthesis. Again, general biology review. I want you to look through this. Any of the large print, you should know what a ribosome is. You should know the difference between DNA and RNA, messenger RNA. So I want you to take some time and go through this plate as a review. So look at the first 4 plates as a general biology review. And now what I'd like to do is focus on plate 5. Metabolism and production of ATP. The energy currency of the cell is adenosine triphosphate. That is used by all cellular processes to drive the machinery, the mechanism of cell functioning. I spent a lot of time on muscle physiology, interested in how muscle utilizes energy, how it utilizes ATP, and from the standpoint of locomotion and energetics, the major energy consumer of the body is skeletal muscle. However, even in an inactive animal, there is substantial energy metabolism. We can express this as basal metabolic rate, that there is some level, some cost to living. Pumping ions, pumping the muscle of your diaphragm to breathe, this all requires ATP. So how does this ATP, where does this ATP come from? And there are two primary sources in terms of ATP sources. And we'll break those down into aerobic and anaerobic. One component of the physiology is anaerobic. So an means without, aerobic means without oxygen. So anaerobic metabolic processes function in the absence of oxygen. This is probably the most evolutionarily the most primitive type of metabolism. And then we saw the evolution of aerobic, or oxygen demanding. So what's the trade-off? If you look on your chart, you see anaerobic, no oxygen, and the substrate that's utilized in the anaerobic metabolism is glucose, or carbohydrates. So it turns out that only carbohydrates can be utilized in the absence of oxygen. The archetypal carbohydrate is glucose. When I talk about carbohydrates or I talk about sugars, glucose is usually the example that I use. On plate 5, you see that 1 mole of glucose do I have a pointer on? Let me do pointer. There we go. 1 mole of glucose is broken down to pyruvate. Now, the problem with pyruvate is the necessity for regenerating, reducing equivalents. When you take biochemistry, you are going to spend a lot of time on the functions of NAD and NADH, electron acceptors. What you need to know for this course is that pyruvate is not an adequate place to stop anaerobic metabolism. Anaerobic metabolism results in the conversion of pyruvate to another compound. In human skeletal muscle, this compound is lactate. Now, it turns out that other anaerobic organisms will take sugars and convert them to other compounds. So instead of pyruvate going to lactate, if you have the right yeast, pyruvate is converted to ethanol. Other microorganisms will take pyruvate and convert it to acetic acid or vinegar. Historically, the drive to study the biochemistry of anaerobic metabolism was driven by the wine industry. Louis Pasteur was hired as a chemist to study why so many bottles of wine ended up as expensive vinegar and not as drinking wine. And it turns out it had to do with the microorganisms that were given the sugar. Now, in the wine industry, sugar comes from grapes. Vodka, the starch is a potato base. Sake, it's rice. So you can see whole industries and large sums of money were tied up with the biochemistry of anaerobic metabolism. The way I'm going to make my fortune is to splice in the genes that instead of converting pyruvate to lactate, convert pyruvate to ethanol. So that when you go out and you exercise very hard, instead of producing lactic acid in your muscle, you'd produce ethanol. I think we would see a huge increase in the number of people that are out in the field exercising. For skeletal muscle, end product is lactic acid. That's a problem. You are producing a metabolic acid. The pH with lactic acid will decrease. A decrease in pH will alter enzyme function and is one of the processes that leads to fatigue. So we find that fatigue is correlated with increased levels of lactic acid. So the more reliance you have on anaerobic metabolism, the more you're going to alter the pH, the more lactate you're going to get, and the more fatigued you are. So what's great about this? I mean, if we're not producing ethanol, why produce lactic acid? This process is very, very fast. That is, you can convert glucose to lactate extremely rapidly. That means you can generate ATP very rapidly. That is important in escaping from the cave bear. If you need your muscles, your muscles need that ATP very quickly for a sprint or a very fast start, that's a process that will provide that. It does so not only at a cost, that is, you're getting lactic acid, you only get 2 ATP. So there's a lot of metabolic waste. You're not getting all the energy that you should be getting out of this compound, but you're getting it fast. All right, so those are the trade-off. You can generate the ATP fast. You can't do it for very long because you fatigue. And you don't generate a lot of ATP. You generate lactate, and the only substrate you can use are carbohydrates. Again, primitive organisms. As we see the evolution, we see the advent of another organelle, the mitochondria. The mitochondria serves in oxygen transport. So if you look over on the right-hand side, you see aerobic metabolism. So in the presence of oxygen, you're no longer restricted to lactic acid. The pyruvate, so here's the same pyruvate that you see over here, but in the presence of mitochondria, you can shuttle that pyruvate into the mitochondria, binds with acetyl coenzyme A, and drops into the citric acid cycle. So again, if you're taking biochemistry, they're going to make you memorize all of the intermediates of the TCA cycle, sometimes called the Krebs cycle, the tricarboxylic acid cycle, or in this case, citric acid cycle, because the first compound that binds with acetyl-CoA, the first compound that's formed is citrate. So it has a number of names, but its function is still the same. Generate lots of ATP. And then you'll see that that the compound in the citric acid cycle generates reducing equivalents that then go into the respiratory chain, and you get out 34 ATP. It's not important that you memorize this number down here. Okay? 34 and 2 is, biochemists make you memorize that stuff, but not for this class. What I want you to know is that with aerobic metabolism, you generate more than 10 times the amount of ATP. So you're getting more energy. What's the waste product? If you look down here, you'll see oxygen is converted to water. Relatively non-threatening end product. Okay, it's not lactic acid. You get a lot of ATP. You can burn sugars. So the example here shows glucose. Aerobic processes also burn protein and fats. So all of the substrates can be burned or metabolized in an aerobic environment, only carbohydrates in an anaerobic environment. Well, if this is such a good deal, why not do this all the time? This aerobic process is slower. So if you are in need of ATP, if the decision between life and death is how fast you get the ATP, anaerobic metabolism is going to serve you better than aerobic metabolism. Plate 6, metabolism, respiration, and the citric acid cycle. All right, now, so see that circle? That's the circle, the TCA cycle, all those intermediates that you're going to have to memorize in biochemistry. You don't have to for this class. I merely show it as a reminder. If you've had this before, this is review. If you're going to have it, this is kind of the premonition that this is an incredibly important biochemical pathway. It generates lots of ATP. It occurs in the mitochondria. So the more mitochondria you have in a tissue, the more aerobic it is. The respiratory chain. So down in the lower right, there's the electron carrier and ATP synthase. When we talk about reducing equivalence to the transfer of electrons, those are used and coupled to the production of ATP. And there are a number of proteins that are embedded in the walls, the membranes of the mitochondria, that pass, if you will, the electron from one protein to the other. And there's a chain of these, so it's sometimes referred to as the electron transport chain, or in your book here, they refer to it as the respiratory chain. But it's the idea of passing these electrons down from protein to protein and coupling the production of ATP. The final electron acceptor is a cytochrome. So it has a name that reflects some of its properties. It is a chrome or colored protein associated with the cell. And the final one of these cytochromes is called cytochrome C. Cytochrome C is a red colored pigment. Cells which have lots of mitochondria have lots of cytochrome C. That lends a red or brown color to that tissue. So in fact, one of the most aerobic tissues is a tissue called brown adipose tissue. It's lipid, but it's brown because it is chock full of cytochrome Cs. The primary function of brown adipose tissue is to produce heat. So now we're going to have to revisit the single most important equation, chemical equation, the oxidation of glucose. So C6H12O6 plus 6O2 goes to 6CO2 and 6 waters. When we talk about waste products, there's water. We also get CO2 produced. This is an aerobic metabolism. It's in the presence of oxygen. And if you're a straight chemist and you don't have biology in your background, all you're interested in doing is measuring the amount of heat of this process. So in a chemistry lab, they might ask you to measure the heat of combustion. And I forget what it is. Yeah, I had to memorize it at one point, but 1 mole of glucose, if you were to oxidize that, you get several hundred kcals of heat. Now, biological organisms trap some of that heat in the form of ATP. But even biological organisms, if they produce ATP, 60% of the energy is going to appear as heat. So that's one of the consequences of metabolizing. The more and faster you metabolize, the more heat you generate. After eating, feel that increased temperature? It's real. You are metabolizing foodstuffs. And in the digestion process entails making ATP, but still, when you make ATP, you make heat. So if you have lots of mitochondria, you have the capability for doing lots of aerobic metabolism. You can produce lots of ATP, but you will also produce a lot of heat. Okay, let's take a break.